Hi, my name is Zelda Najwa binti Razali with matric number 181768.
Hi, my name is Ko Hui with matrix number 184062.
Hello, my name is Nona Suha binti Muhammad Noh with matric number 184145.
Hello, my name is Nurul Izzatul Nadia binti Zulkafli with matric number 181787.
Hello, my name is Yong Chen Hui with matric number 184901. Okay, today we will discuss about storage disease, which is mucopolysaccharidosis in goat. Okay, actually this is lysosomal deficiency disease, and the learning objective of our discussion is to identify the sign of the mucopolysaccharidosis in goat, and next to identify the cause and diagnosis of mucopolysaccharidosis in goat, and third to determine the failure in biochemical event and processes which cause the mucopolysaccharidosis in goat. Okay, who can explain to us what is the cause of mucopolysaccharidosis in goat?
Okay, I can explain to you all about the cause of mucopolysaccharidosis in goat. Okay, firstly I will talk about the general. Okay, in general, mucopolysaccharidosis are a group of metabolic disorders caused by the absence of specific enzyme needed to break down molecule called mucopolysaccharides, or also known as glycosaminoglycans, which is GAGs. Okay, thus it will make the mucopolysaccharides or GAGs deposited in their body tissue. So that is in general.
Okay, and I found something like more information about glycosaminoglycans or GAGs. They are long sugar carbohydrate chain found in numerous cells in the human body. They perform a variety of functions. Under normal circumstances, the body routinely will break down the polysaccharides to prevent the overabundance, and enzymes typically metabolize these complex sugars at the end of the cell's lifespan. The presence of certain genetic disorder known as lysosomal storage disease render the body unable to perform this process. So the researchers studies the glycosaminoglycans and their function in various disease processes. The polysaccharide building blocks connected by other molecules are performing thousands of chain variations make up the glycosaminoglycan. The majority of glycosaminoglycan in the body are non-protein hyaluronic acid, while others are including chondroitin and heparan sulfate molecules. These glycosaminoglycans play a large part in cell development, repair, and replacement of cells, and the chain are vital constituents for the development of many systems in the body, including corneas, cartilage, and tendons, skins, and connective tissue. So means that actually the glycosaminoglycan is a very important component in our body, because it is like play the main role in the development of the system. But may I know how can this disease related to goat?
Okay, so for goat, which is the small ruminant, also acquired this disease because of this disease is genetic defect, inherited disease that can cause fatal to the goat. Okay, so this genetic defect is known as mucopolysaccharidosis double 3D. Okay, triple 3. Mucopolysaccharidosis 3D. Alright, okay. In another name, it is also called as G6-sulfase deficiency, and it is usually referred to as G6S. Okay. So, caprine mucopolysaccharidosis result from the nonsense mutation and consequent deficiency of lysosomal and acetylglucosamine 6-sulfatase activity that associated with tissue storage and urine excretion of heparan sulfate glycosaminoglycans. Okay, glycosaminoglycans, which is GAGs. Okay, so using special stain, the mucopolysaccharides were identified in most tissue of affected group. Okay, so that's all I know about the cause of this disease. So, may I know what is the history of this disease?
Okay, I can talk about the history of disease. Actually, this genetic disease in goat is first defined in Michigan State University in 1987. The researchers from the Michigan State University had concluded that about 25% of Nubian carry this genetic disease. Nubian is a type of goat. Nubian is a breed of domestic goat, which also known as Anglo-Nubian goats. This genetic disease happened in the goat as a result of single mutation and appear to be confined to Nubians and their causes. The affected goat that lack in the specific enzyme to break down the mucopolysaccharide will result in a variety of symptoms of varying severity. That means that this disease is first found in 1987, and they found out that there are 25% of Nubians carry this genetic disease, and this also affect the others goat which carry the Anglo-Nubian goats genes. So that's all for me. And can I know how about the sign? Because there are some symptoms that show that they get this MPS 3D, right? Who can explain to us about the sign?
I will explain about the sign. So this mucopolysaccharidosis 3D disease can be observed in central nervous system of affected goat. The morphological change can be observed in the central nervous system, such as neurotic expansion and other neural alteration that may have functional significance were also seen. So this disease also caused the spectrum of lesions were greater in the severe form CAP3 mucopolysaccharidosis 3D and included mild cartilaginous, bony and corneal lesions. The more neurological deficits in the severe form were related to the greater extent of the central nervous system. This myelination, where the myelin sheath of neuron is damaged. The physical symptom that can be seen exhibited by affected goat is failure to grow. So failure to grow, so it cannot grow. So it doesn't get big. Other physical symptoms include deaf or blind caused by compromisation of immune function. So it's deaf and blind and small because it cannot grow.
I think cannot grow means they will die at young age.
They will die at young age. The affected goat lack in muscle mass and appears lopsided and sometimes with bulky head. This genetic defect will cause fatal, especially to the baby goat, and cannot live longer.
Okay.
So it will die before it can grow. It will also be deaf and also be blind.
This is okay. Okay. Then where I know the diagnosis of this disease?
Okay, so I will further explain about the diagnosis of this disease, mucopolysaccharidosis. So the disease actually can be accurately diagnosed only by the identification of the point mutation by genetic analysis. Recently, a gel-based polymerase chain reaction (PCR) assay and a subsequent LUI restriction enzyme digestion was shown to differentiate the genetic form of this allele combination. Similarly, the same approach has been used for the identification of Sanfilippo syndrome. In the present study, a real-time PCR single nucleotide polymorphism (SNP) assay was developed to facilitate accurate diagnosis of this disease, that is mucopolysaccharidosis 3D.
Okay, I heard that you talked about the PCR. May I know what is PCR?
Okay, PCR or or it's called also polymerase chain reaction is the amplification of a specific DNA sequence, term target or template sequence that is present in a complex mixture by adding two or more short of oligonucleotides that also called as a primers that are specific for the terminal or outer limit of the template sequence. The template primers mixture is subjected to repeated cycle of heating to separate the double-stranded DNA and cooling in the presence of nucleotide and DNA polymerase such that the template sequence is copied at each cycle.
It's like copy the DNA so that we can do something on it. It's like that. So in general, that means that the diagnosis we can diagnose this disease by using PCR. Yeah. Then how about the chemical event? Because when there are some disease occur, that means that in their chemical processes there are something wrong with it. Who can explain to us what's wrong with the chemical processes?
Yeah, I can further explain to all of you about the chemical event that occurs here. Okay, first, MPS 3D is one of the rarest MPS syndromes which characterized by the deficiency in the lysosomal N-acetylglucosamine 6-sulfatase 6X activity. In the naturally occurred caprine MPS 3D model, the deficiency of 6S is consequence of C322T nonsense mutation. When the 6S activity is insufficient to desulfate the alpha-linked N-acetylglucosamine of heparin sulfate HS, the lysosomes of most cells will accumulate the uncatalyzed HS gas. The resulting accumulation of HS gas in neurons, astrocytes, and cells of some systemic organs induce a secondary storage of glycolipids, partly through effect on activity.
May I know what is astrocytes?
Okay, good question. I'll explain what is astrocyte to you. Astrocyte is actually a neuroglial cell of ectodermal origin, characterized by fibrous or protoplasmic processes, collectively called exoglia or microglia.
Okay, thank you for the explanation.
Yeah.
Okay, I found something about the neuramidase. It actually is a hydrolytic enzyme that occurs on the surface of pneumococcus influenza causing virus and some paramyxovirus and as an antigen that speak mucoprotein by breaking the leukocyte link. It means that it's an enzyme that found in the something bad like virus and something like virus. Okay, it's something like neuramidase, neuramidase, but it also formed in others, like it's not all bad, but there are some good because some are in our body.
Okay, I will give you my explanation. The heparin sulfate classes of sulfated glycosaminoglycans link to serine residues in core proteins through a common tetrasaccharide construct. This process is initiated by the xylosyl transferase catalyzed attachment of xylose using UDP xylose as donor. After the attachment of the tetrasaccharide, the proteoglycan structure diverges since the next carbohydrate linkage determines the glycosaminoglycan class attached. Alpha glycosidic bond to N-acetylglucosamine results in the attachment of heparin sulfate.
Sorry to interrupt you again.
Yes, question again.
So what is proteoglycan?
Okay, a good question.
Okay.
Okay, that means you listen patiently to me.
Okay.
Okay, proteoglycan is any of a group of glycoproteins found primarily in connective tissues and form of subunits of glycosaminoglycans linked to a protein core. Hydrated proteoglycans form the highly viscous fluid of mucus and a matrix of the intracellular ground substance of connective tissue. So it's also called mucopolysaccharide.
Okay, so I will add on healing points. Okay, actually the 6S activity is also needed to desulfate the beta-linked and acetylglucosamine residue of keratan sulfate. However, beta-hexoaminidase is able to act on the non-reducing end to produce an acetylglucosamine 6-sulfate monosaccharide that will become the storage product and representing keratan sulfate. In addition to the neuronal colocalization of HSGAG and granulocytes in mucopolysaccharidosis 3D, intraneuronal accumulation of alpha-beta-1 40 peptide has been documented in the mucopolysaccharidosis. So the role of the alpha-beta peptide and the uncatabolized HSGAG and also glycolipid substrate in the neurodegeneration and profound dementia of mucopolysaccharidosis remains to be elucidated.
That means that there are two process. Degradation of heparan sulfate. One is degradation of keratan sulfate. But because the lack of enzyme, what is the name of enzyme?
G6S
or G6S. Okay, because lack of G6S, so the event cannot occur, right?
Yes, you're right.
And I found out actually it's because there are mucopolysaccharidosis in goat, but actually there are many types of mucopolysaccharidosis. And for example, like Hunter syndrome is also a type of I think the Hunter because somewhat the name of the developer of this disease. It's not it's like Hunter syndrome is mucopolysaccharidosis 2 actually. Two only. Okay. This is because the lack of this is because there are some also it's the same like our case is also has some deficiency in the enzyme, and so that the iduronate sulfatase cannot be converted into alpha iduronidase, and there are many others like Hurler syndrome and Sanfilippo syndrome A, and which also known as MPS 3A, and many more, but it's not occurs in goat, maybe in humans or in other animals. And here is some diagram I found in internet. Actually, this is the heparan sulfate degradation, and actually the causes of this disease is occur at the end of this chain. That means end of the process when the beta glucose is N-acetylglucosamine 6-sulfate, right? Just now we mentioned that it cannot convert to the next product because of the defect in the enzyme. And the keratan sulfate is also the same. It's also formed at the almost the end of the process. It's also N-acetylglucosamine 6-sulfate. Sulfatase cannot convert to the beta-hexoaminidase AMP. So that means there are only two defects in this two, but for the overall of the mucopolysaccharidosis Mucopolysaccharidosis. Actually, there are three, but I forget another one degradation. It's also like glycosaminoglycans degradation, but in that process, the MPS 3D is not occur in that process, so we don't discuss about that. We only discuss about the two heparan sulfate and heparan sulfate. Okay, that means can we do some conclusion about this?
Sure. Okay, so I will first conclude about the cause of mucopolysaccharidosis 3D. Okay, so in general, it is because of absence of specific enzyme, which is mucopolysaccharides, but in goat specifically, it is result from the nonsense mutation and consequent deficiency of lysosomal and acetylglucosamine and sulfase activity. Okay, so that's the causes.
Okay, and how about the sign?
Okay, the physical sign that we can see is the failure to grow, deaf or blind, lack in muscle mass, and sometimes with broken head. That's all for the physical sign.
And one is maybe better, right? Maybe better.
No, like that more.
But just now you mentioned that maybe the goat may die because of this disease.
Yes.
What I have learned is this disease is first found in 1987. And then it's from Nubian goats.
Yeah, like you want the
25% of Nubian goats carry this count.
Yeah.
Yes.
So I will conclude about diagnosis. Actually, this disease can be diagnosed by two ways. First, first a gel-based polymerase chain reaction, PCR assay, and also subsequent LUI restriction enzyme digestion. And this assays also can be used to identify Sanfilippo syndrome.
Actually, Sanfilippo is another name of mucopolysaccharidosis. Actually, we feel because it's at that very days, they sometimes they call them with that name just mentioned Sanfilippo, maybe mucopolysaccharidosis or MPS, and sometimes it's also known as GCS deficiency. Okay, I think we all know all about more about this MPS disease. Okay, how we can get more knowledge on this. Okay, I think our discussion ends here. Okay, thank you.
Thank you for the last discussion.